I'm Ellie Nedivi. I'm a professor of brain and cognitive sciences, and I have an appointment in biology too. And what I'm interested in is the brain's ability to constantly adapt, what we term as plasticity. And it's a fundamental feature of the brain to be able to constantly change as need arises. And even though we study it because it's such an important aspect of brain function, it turns out that when things go wrong, many of the things related to plasticity are the culprits. And the other thing is that if we are thinking about damage or any sort of degenerative processes or even disorders that are genetic, if we know more about how the brain itself can adapt, we know what the points are where we can intervene to help. So I think that even though we work on questions that are really basic science, it turns out that there's a lot of interface with potential therapeutics, even sort of predictive assays. I actually am excited about all the projects in the lab. So there are kind of two directions that we have in terms of, even though our overarching question is related to brain plasticity, one is using imaging as a tool to look at how connections are made in the brain in real time, where we can label neurons or brain cells and the connections between them, which are synapses, and we can monitor them coming and going over time and things that influence that. So that's an incredibly powerful tool to look at plasticity in the living mouse brain, which we're doing. And so and many very basic features of how that happens have sort of revealed themselves to us through just being able to visualize the connectivity and how it, and track it over time. So because of that, all the projects using the imaging as a tool, I think are super exciting and kind of discovery projects. Which is why I love them, because we keep finding new things, some of which we weren't really looking for or weren't expecting, and have allowed us to really gain mechanistic insight, even though what we're doing is just observing. So that's kind of one direction that I think is super interesting. So there are different types of neurons in the brain, and there are different types of connections between cells that are of different types. And what we're seeing is that not all connections are created equal. There are some connections that are very rigid and non-flexible, sort of hardwired, you might say. And on the other hand, there are some connections that are very plastic and dynamic. And so it seems that not all parts of the circuit are equally amenable to plasticity. And this is something that we're seeing for the first time, because we can actually track dynamics over time and look at the connectivity and see how it's changing and who it's changing, and kind of the specificity of those types of changes. So that's kind of, in general, one thing that we've been able to see, that there's a lot more heterogeneity. And obviously, if you're interested in plasticity, your ability to intervene depends on your knowing what parts of the circuit are amenable to change. And that's where you're more likely to be able to make a difference.